Welcome to MACSMIN2025. Our next talk will be by Dr. Olga Anosova, who will talk about the complete and bicontinuous invariant of protein backbones. Over to you, please.
Okay, thank you very much, Vitaly. Hello, everybody, and thank you for an opportunity to present here invariants of protein backbones. And since I think most of the audience here do not deal with proteins routinely, I will start from the very basics, that is from the definitions of the proteins. So the proteins, the building blocks of proteins are amino acids. And for the typical proteins, there are 20 of those, which you can see depicted here quite briefly. What is common in them is that actually each of the amino acids is composed from just 4 atom types, which is oxygen, hydrogen, nitrogen, and carbon. Well, and then when they combined, lots of interesting life forms appear, well, which makes us all think about combinatorial explosion and things like that. So it all starts with just 4 basic atoms, COHN, and then they are combined in different combinations into approximately 20 amino acids. And here's one of the simplest one, the glycine, depicted briefly, schematically. And then those amino acids are connected to each other through so-called peptide bonds when nitrogen is connected to the next carbon atom. Well, one of the oxygen atoms is dropped. And then, well, water is not here, as Michael correctly mentioned. Hydrogen is a very tricky thing to catch and observe. So I do not even depict any situations. So what happens is that by a peptide bonds, carbon is connected to the nitrogen of another amino acid, and then we will have a dipeptide, and then another amino acid is connected in a similar fashion, and then we have a longer and longer chain of amino acids. It is also called peptide chain or protein chain. And a protein chain formed from amino acids will consist of certain residues, and each residue is a single amino acid. So it turns out that the question how to connect them in practice is even trickier than the question of how to combine basic atoms into amino acids. And that's basically the question connected to the famous task of protein folding. So it turns out that it's much easier to determine the sequence of amino acids, so-called primary structure, that is connected together, than to actually define what will be the geometric shape of their connection between each other. So the geometric shape of the whole structure, well, starting from the chain and then combination chain, is called a tertiary structure. So we start from the primary structure, just the sequence of amino acids, which are often denoted by letters. And from just the sequence of amino acids, we would like to find the geometric information with all the possible details of the 3D structure. And that is the problem that deserved a Nobel Prize for chemistry a couple of years ago. But we will try to explain what is still left in this problem and how to tackle it systematically. So here we start with what is inside of each peptide chain, each protein chain, and that is so-called protein backbone. So protein backbone, it is just a collection of the nitrogen, so-called alpha carbon, one of the carbons, carbonyl carbon, another carbon, and well, that's it. And this collection, which is connected into a chain, is known as a protein backbone. Where on the third picture, you will see even a simplified version, which just has carbon atoms in there. But it's probably a bit too simple. Why I include it here? Well, because it's a nice structure and there's a new name alpha helix in here. So to help us to deviate from this quite complicated structure in here to something simpler, people tried to also fill this gap by considering so-called secondary structures. And the secondary structures will be covered in way more detail by Jack, who will present in one of the next topics. And basically, there are just three types of helices, sheets, and everything else. So that's the overall picture. Well, quite often people just deal with something like that. Those are the helices, those straight sort of bits of line are the sheets, and everything in between are just the loops. But I like to call those pictures, which we very often see instead of anything else, an artistic representation. Because they are heavily smooth, well, you can compare that sort of more hedgehoggy picture in here with quite a smooth and nice representation there. And I just wanted to demonstrate also how in practice this backbone could look like. So I will try to switch from this window into another window. So here is the exactly same, believe it or not, chain that was in the previous picture, but that's how it looks in practice if we just connect all the coordinates in here. So, well, can you see any helices? Yes, they are there. Wonderfully enough, they are maybe not as smooth as before, but they are present. Yeah, so you can see one, for example, here. Yeah, so and we can sort of check that it's indeed a helix. You can see now the center of it and circle around. So they are present. So those structures are indeed there, but they are never as smooth as on that typical pictures we typically deal with. That is just, I think this kind of pictures is not something many people look at, but I think it's quite a nice demonstration of, well, variability of representations, even of very simple objects, even like helices. So going back to my slides, is it control? Great. So that is the same structure. Back to the backbone. So we saw the pictures of the backbone, a couple of representations. We are mathematicians. That's why we use our own notation to the common representation. And let me remind you that we had two carbon atoms. One was quite crucial in connecting to the nitrogen and creating the peptide bond. And another one was in the middle and had all the side chains which we omitted so far. So we introduce different notations for them, so-called alpha carbon, which is not connected to the next residue we denote as A, and another carbon that is used in the connection we denote as C. And well, then the backbone for us will consist of a collection of nitrogens, alpha carbons, carbonyl carbons. So how to actually consider this shape? And again, as always, even for proteins who are very, very non-rigid, we enjoy considering rigid motion. Well, why does it make sense? Well, I showed you the picture of an enzyme, and one of the important hypotheses by Ronald Fisher stated that enzymes and proteins interact with everything else when the shapes match as lock and key mechanism. probably you remember those explanations of lock and key mechanism from COVID virus when those spikes were the keys into existing locks in proteins. That's why it's important to be able to recognize the shape of the key and the shape of the lock. Well, actually, the more complicated or more up-to-date hypothesis is so-called induced fit hypothesis, which states that actually the key can be slightly different shape, and the locks can also be ever so slightly different, take the shape of the key. That's why, yes, the rigid motion is the strongest equivalence to consider, but ideally we would like to also be able to trace small deviations from those rigid motions. But again, let's start as mathematicians from the very basic equivalence, that is equivalence of rigid motions. Well, okay, now we would like to consider it on the, we can consider different equivalence classes. Yeah, equivalence also appeared in the previous talk. Let's think what kind of equivalences we can consider, at least theoretically, for the proteins. So the simplest type of equivalence will be equivalence by length. Yeah, so if it's the same length, we can call them equivalent. Well, does it make sense? Well, not really. And the first issue is that, yeah, it's not much information. And second issue is, in practice, quite often we do not have measurements for the whole chain, just for the part of that. But it is possible and it's manageable and it's quite doable because then we are just dealing with one single length. If we just considering the sequences, then our possibility of equivalence classes is much larger. It's up to 20 to the power of m. But if we start considering all possible geometric conformations in 3D space, then, well, we are dealing with the infinite dimensions. So how can we still distinguish between rigid classes in the infinite dimension? Well, again, that's the same topic that will be, I think, throughout the whole day mentioned here. That's the theory of invariance. And I will remind the first speaker that invariant is just a property that is preserved. In our particular case, that's the property that is preserved under rigid motion, because that's the property that we are mostly interested in. In practice, what does it mean? It means that invariance offers a tool to quickly find different objects, which means that if the invariants are not the same, then we can quickly say that, oh, the objects were not the same by rigid motion, rigidly different. But as mentioned again in the previous talk, if you would like to say that coinciding invariants indeed mean coinciding objects, we need a stronger notion than just invariant. It is a notion of so-called complete invariant. Okay. Well, any simple examples of an invariant for protein chains? Just an invariant of a protein chain. What can be the simplest invariant possible one can think of?
Chain length.
Great, perfect, a very nice one. And that's indeed a very simplest one. It is mentioned, yes, it is an invariant, but not very informative, because many different chains, even with different amino acids, can have the same chain length, which means an invariant. But if it is an invariant, but it's not very interesting. It's not a complete invariant. Well, another common invariant used in biology is the torsion angles. So, I think one of the next speakers will cover that in more detail. But basically, the torsion angles, that's just two angles out of all possibility of parameters. And how many parameters we'll need, we will see in a moment. Which means that torsion angles indeed are handy, are invariants, but unfortunately they are not complete invariants. They do not create the full picture under the rigid motion.
How about chirality?
Chirality, you mean the, well,
Left to right.
left and right. Yeah, so we are considering rigid motion, which means rotations and translations, and we are not dealing with invariants of isometry yet, which means that we should be able to differentiate the chiral proteins, which I think is quite important in live structures. So yes, we would like to be able to definitely find them out. But thank you for a very nice question. Okay, but as mentioned, also, yes, rigid motion is nice, but not enough even for the induced fit lock mechanism, which means that we would like also to have something to quantify small dissimilarities, or not so small dissimilarities, which means that for proteins, it's particularly essential to have distance metric. And again, well, just reminding from the previous speaker, here is the definition of a distance metric. That is the function that satisfies the following three axioms. And yes, the existence of invariant or even complete invariant does not automatically means that we'll have a nice metric. Firstly, well, the trivial metric is something which will be 0 for coinciding objects and 1 for non-coinciding objects. That's something we all deal with when we do some classification. Yes, it's binary yes, no, equal or not equal. But it does not allow us to measure small deviations from this equality or equivalence. That's why we would like something stronger. We would like not only a distance, but we would like to have a continuous distance or continuous metric. So now I'm ready to state the method. We would like to find a map on an invariant from the backbones on all the backbones that will allow us to differentiate them under rigid motion. In particular, we would like those invariants to be complete invariants, so notice if and only if here. So we'd like to find complete invariants for protein backbones, but also, well, I've mentioned one strange discontinuous distance, which is practical but not very meaningful. It turns out that there could be, mathematics is rich enough to construct lots of different complicated things which will be not very useful in practice. That's why one of the important requirements is that any backbone should be reconstructible practically from our invariant. And that is also not a given. And yes, as mentioned, we would like to have a metric. We would like the metric to be continuous, more precise, Lipschitz. We would like to have Lipschitz continuity, which is one of the strongest continuities we might have. Well, basically, it means that if two objects are not very far from each other, very close to each other, then we would like to have the same property for the distance, for the invariants, that invariants should be not very far from each other in the chosen metric. And also we would like the opposite to be true, which means that if invariants are not very far from each other, then we would like to be able to state that initial objects are also not far from each other. That is so-called reverse continuity. And again, the strict formulation is here. And we are dealing with practical problems, which means that, yes, computability is a very, very, very important factor. So we would like all our algorithms be linear time. Okay, so the main problem statement is on this slide. Any questions here? Great. Okay, so we will move on, but if any questions appear, please do ask. So how do we solve this problem? We solve this problem by introducing so-called backbone rigid invariant. So how do we construct it? we take a chain of M amino acids or M residues. We take the backbone of those residues, and then we start from the first residue and introduce the bases in this residue. And then every next part of the backbone from the next residue will be represented in the coordinates introduced in the previous residue. Well, more precisely, in the first residue, we take N nitrogen, alpha carbon, and carbon, introduce the orthogonal bases with the center at alpha carbon, which also were strategically chosen to allow possible extensions to take into account side chains. And then the next residue will be written in the coordinates of the previous first residue. Well, more precisely, we take the three vectors which actually form the snake that you've seen on my representation of the backbone, and each of the vectors is represented in a coordinate system of the previous residue. And the same process is repeated again and again from one residue till the next residue, till the next residue, till the next residue. As a result, we will have M times 9 matrix. Well, M is the number of residue, why 9?
Three things, the three coordinates.
Absolutely. So 3 atoms in the next residue, or well, 3 vectors connected to them, and 3 coordinates per each of them. So very simple logic behind it. Okay, and then we claim that this matrix or invariant solves our problem. That is, it's complete, and well, all the other properties as mentioned. Well, whilst you might be thinking, so what, if I'll do some hand waving here, because I think it's important to demonstrate why we did what we did. So basically what we did, we took a chain, and a chain can be seen as a sort of a snake, yeah? And the problem, as always, as mentioned yesterday and today morning, is that if you just write the coordinates in some strange coordinate system, then it will be hard to compare different snakes with each other. So the question is how to create the coordinate system that will be somehow connected to the snake. And the very first idea is, well, just introduce the coordinate system in the very first residue of the snake, and then describe the whole snake in this coordinate system, right? Any downsides of that method?
Potentially propagates an error early on.
Yes, protein chains are very long, as you've seen in the pictures. Well, that picture was not the longest one, which means that the error will propagate. But what we do here, we represent the next residue in the coordinate system of the previous residue, thus negating or minimizing those possible errors, and we stop them from propagating. And here the theorem that was proved and published is that indeed this BRIE completely solved the stated problem. Well, I've promised distance. So the distance that we've considered is the L infinity distance, which allows to quickly compare different shapes, rigid forms. So L infinity is just the maximum absolute difference between them. But also it can be handy to use root mean squared, and actually all considerations are repeatable for root mean squared, but we've formulated and proven everything for L infinity. So for L infinity and the invariant, well, firstly, as mentioned, everything is quite fast. That's exactly what we needed, and how fast I will demonstrate on a practical scenario. Now, why I didn't like the length of chain as an invariant, because as mentioned, yes, quite often in practice, we don't have the full measurements for the full length of the chain. Yeah, the chain is long, and we could have restricted ourselves just to parts of it, which means that in practice, it is very important to be able to compare the chains of different lengths. And additional property, which I did not formulate before, I should say, but which I would like to highlight here, is that this invariant respects subchains. Or what does it mean? That means that in practice, either subchain has the same invariant, exactly the same invariant as part of the longer chain, or, the possible exception is the first residue, but again, the first residue of a shorter chain, if it's not from the very start of the long chain, it can be easily reconstructed. So that's sort of an additional property, which I would like to highlight, but which was not formulated in my initial statement. Okay, so what I did mention and did promise is Lipschitz continuity, and indeed it happens to be true for L infinity. Indeed, if our objects, our chains are not very far from each other, then invariants will be close to each other in L infinity metric. And it turns out that the opposite is also true. That is the inverse of Lipschitz continuity. Well, you can notice that coefficients are getting quite nasty in here, but still, if we have the Brie invariants close to each other, then we can measure and state that initial objects are also relatively close to each other, and those variables they can be, that's an upper bound, which means that in practice it's quite sufficient. Okay, so that was theoretical achievements. Now let's see how those are applicable in practice. And in practice, as again was mentioned in a previous talk, the largest collection of proteins, so-called protein data bank, is considered to be a gold standard of experimental structures. So each protein has a 4-symbol code. Well, there are some examples which correspond to the 3 pictures below. And then each entry also, each file can have different versions of model, can have different molecules or entities and different chains. So in essence, every experimental protein is quite a complicated construct, similar to what you see in front of you. So the 3 structures in front of you, what do you think? Are they same, different, similar, not similar? Just your impression.
Look very different.
Oh, they're kind of spherical.
Can't tell, right? Yeah? That's probably the correct answer. Can't tell. Well, yeah, that's artistic representation, and I can tell that it's quite a complicated entries. Well, not the easiest ones, which means that, yeah, can't tell. And especially, well, if you would like to look inside, again, it's quite complicated, right? So can we say anything about those complicated structures? So we've started with just the entries, 230K. If we look inside the entries and separate them into the chains, then there will be more than that, obviously. And we cleaned the chains by removing or filtering out some of the ones that we either didn't like, for example, with very small distance between atoms, which probably means atomic clashes, or very large distances. Also, we excluded so far disorderly chains with varied occupancy. We can deal with that later. And also we removed so far non-proteins to have the cleanest possible database to start with. So by this filtering, we were left with 700K clean chains. And the idea was to compare those chains with each other and see how many classes of rigid equivalence. Well, firstly, computability. It turns out that it is possible to compare all those backbones in about 6 hours on a modest laptop, so we didn't even use HPC facilities. It turns out that invariants are identical in 13,000 pairs. And since they are complete invariants, that means that the pairs can be exactly matched by rigid motion. Yeah, that's exactly the same geometric structure. Well, further, after we filtered that out, it turns out that, well, slightly under 10,000 pairs have absolutely identical XYZ coordinates. And 700 such pairs were also in different PDB entries. That was quite surprising.
These are all chemically different though.
Well, I'll come to that. That's a very good question. Yeah, everything in the world is related. Yeah, so your question is very meaningful. Yes, a very good question. I'll cover that. But the whole idea, the whole number and variability of the results is quite surprising. And let me notice that here it's the complete coincidence. Yeah, that's not even just closed shapes. That's completely coinciding shapes, and sometimes even completely coinciding coordinates. Okay, so we started to look at that in detail. So, for example, here in 9 pairs, we also have found that the sequences of amino acids were different, which seemed to be physically impossible if we claim that the coordinates are exactly the same. Yeah, so that's sort of experimental structures, quite flexible structures, which means that have total coincidence of the even coordinates is quite surprising. And for those, for example, 2 chains, they are quite lengthy, 430 amino acids in a chain, and 9 different residues or amino acids, which is surprising. And indeed, the author accepted the duplication of those 2 entries, well, and said PTB entries apparently can multiply on their own. Well, yeah, and praised our geometric comparison, which allows to quickly identify errors. Well, those are just quite some examples, yeah. And well, here example again, there's just one difference, glycine and blue. I don't remember exactly the name. Just one is different, but coordinates are the same. Well, okay, mutation happens. Does it really mean that the experimental structure will have exactly the same coordinates? Well, not sure. And those 2 are also interesting. Well, length for modest, number of different acids is quite striking. Yeah, so why do they coincide? Well, they happen to be from those horrible creatures, 3 of them that you saw before, believe it or not. So that is just subparts of them, just some chains of them indeed happen to be coinciding. Well, it turns out that they are coming from closely connected by different families of viruses, and they are known to be different even in interactions with receptors and so on and so on, and also in clinical representation, which means that despite their sort of known similarities between the viruses, they are and should be different. Well, how different are they? Can you guess which one? So one of them belongs to this CVB3, one Jew, and another one belongs to 1M11. Can you guess which one which? Well, I can't either. As mentioned, they totally coincide. Well, interestingly, I didn't put the picture for the first one there because we contacted the PDB validation team and they quietly updated the geometric coordinates for the first one, well, not for the second one yet. Okay, so that was a promising start, and then we caught attention of human experts in biology. in protein structures of Alex Vladava and his team. And they did a brilliant job of discussing of the duplicates from the biological point of view. Well, we had quite a few of them, which means that human experts will struggle to look at them all at once. So we took only the very best structures from X-ray representation, from quite a nice resolution demanded by them, from different PDB entries to ensure that, yeah, we are not sort of comparing the same chain turned over again and again. Also, our colleagues mentioned that analysis of viruses and ribosomes is not very practical due to complexity, which means that unfortunately those horrible three structures are a bit tricky to deal with by a human expert again. So yeah, we found those situations. So the final data set to consider by a human consisted of 56 pairs. And yeah, well, the idea was to identify possible cases and maybe call this to maybe have some recommendations to the PDB team or to others how to deal with them, what to keep attention to. Well, one of the group was subsequent redeposition or in the PDB. So that was the situation of those duplicating PDB entries which appear by themselves, yes, confirmed by the author. Well, those structures are still in the PDB, and it turns out that Vladava and his team are also picked up on them for various different reasons. Well, but the structures are still there, still not corrected after a couple of years. Well, there were structures which were deposited very close together, again, to the question of how they're connected. Yeah, so there were structures that were deposited with a one-day difference, and maybe they've corrected something, but still both structures are there in the PDB. None of them is removed. Well, also there were cases when redeposition should be different by description. For example, yes, mutation of a single one or larger cases, but still coinciding by fact, which sometimes indicates potential non-experimental modeling, shouldn't be really the case in an experimental database. Or sometimes the structures were quite clearly refined by removing some atoms that did not fit well into other measurements. Yeah, for example, into R factor. So the new structure has some, well, better parameters, or similarity measures or other parameters, but probably that's not exactly how it should happen. And well, quite a substantial part of so-called, beautifully called other peculiarities. Well, just one example there, different unit cell parameters, but totally same atomic coordinates and other factors as well. Okay, so just one example. So you see, just under 60 situations or cases considered by human experts, whilst our tools are capable to quickly locating and finding thousands of them. And we were considering, yes, so far, just the total coincidences of the coordinates because there were so many of them, but actually there are many more near duplicates. So here you can see primary sequences being identical in green, and here how they correspond to each other by the distance. So you can see that here they have sort of the similar rigid shape, but sometimes the primary sequences are different and still the rigid shapes are very close to each other. Yeah, so should it be like that or not? Well, I don't know. That's something to look into further. Why it was not found out before? Well, traditional tools have striking limitations. For example, well, the typical ones are either LDDT or PLDDT, used by AlphaFold, or TM score. Unfortunately, none of them conform to the metric axioms, and that check started as a MSc student project, and now it continues, and I hope the results will be ready pretty soon. But basically, yes, even symmetry axiom fails and sometimes fails quite significantly. That is not what many people expect at all. Root mean square deviation is a metric, and it is a very good measurement, but not on large data sets, because on a large data set like a PDB is just unmanageable. It will take, well, the billions of days or even years. Which means that our invariant BRI provides sort of geometric style coordinate representation of a protein backbones, which is easy to compute and to compare. Well, just a bit on a positive note, yeah, hopefully those invariants can be useful to open up the AlphaFold box, because now we can compare AlphaFold predictions with all other PDB experimental structures, including just taking the clean structures, not everything as before, which means that, yeah, we can avoid unnecessary repetitions, which inevitably skew machine learning and other methods. So what we see here is just one example of the third most frequent sequence. I've chosen it because it's one of the frequent ones, but also it's relatively short with 20-something amino acids. Here is the picturesque representation of AlphaFold predictions and in gray experimental structures for the same sequence. Well, that is not our invariance. That is the mentioned angle, dihedral angle invariance. Two of the typical one, phi and psi, and another one. And actually that picture, I've included it to represent the possible difficulties in considering the angles. You can see how the picture here, it jumps. Any idea why? What is depicted here? So the hint is that it's an angle, and another hint, well, it has minus 200 to plus 200. That's the student's representation, not the best. Actually, it should be from minus 180 plus 180. Yeah.
Same angle.
Yeah, which means that most of the situations of this angle, that's just a straight line. Yeah, just measured either this way or that way. But if we blindly consider those angles, here we can see that those jumps, which is discontinuity, which in practice means that it's not so easy to compare them if we don't know what to do. Yeah, we can try to shift sort of the scale in here, and it might help in this case, but not completely. Yeah, which means that, yeah, that's the downside of dealing with the angles by themselves. They are well connected to our understanding of geometry, but they can be discontinuous. And I think Will might say a couple of words on that in his next presentation. Next dot. And well, our BRI invariant is continuous as well, again, briefly depicted here. And now we can compare alpha box predictions with the existing experimental structures cleaned by us, and have hopefully some deeper insights than just PLGDT measure, which basically usually just shows some deviations on the ends, and in this particular case, not in the middle.
Sorry, are the grey ones, are they different models of this?
Grey ones is experimental PDB structures.
Yeah, okay.
And we have lots of them because I've chosen the sequence that is in the PDB present in the PDB quite frequently. Yeah, so okay. Approximately 2,000. Okay, so grey experimental, red a single AlphaFold prediction. Right, so yeah, that is just a work for the future, opening the AlphaFold box, and that was just a hint of how we could approach that. But let's do the summary of what was done. So the path similarities that are routinely used, they are useful in some cases, but they unfortunately not conform to metric axioms, or they're quite slow, which means that we needed some other tool that allowed us to compare the geometric structures of existing large data sets And in pairwise comparisons, it is useful as well. And also for better comparison, we've introduced not only complete invariants, but also the continuous metric to trace the differences between those rigid conformations. And here just a quick highlight of one of the next talks. I've mentioned secondary structures. The similar approach to continuous secondary structures will be represented later by Jack. That's all from me. Thank you very much. Any questions?
Thank you, Olga. Let me stop recording.